we scroll down right here, we can see that in our brain is a mass of cells, yes, but we are going to have some cavities, right? We said the brain, the spinal cord is going to be composed of these cells called neurons, as well as the neuroglia. But the brain is not completely full mass of cells because we're going to have cavities. And these cavities are the ones that you see right here in green. These cavities are going to be called ventricles. The first two and larger ones that we have are going to be the lateral ventricle. This is one lateral, the first lateral ventricle, and this is the second lateral ventricle. We have other ventricles, which is going to be this one. This is going to be the third ventricle. And another one right here, which is going to be the fourth ventricle. There's going to be communications between these ventricles. And for example, right here, we're going to have the interventricular foramen. This one is going to be the cerebral aqueduct. And right here, we're going to have the central canal that is going to take the fluid down into your spinal cord. So the cerebrospinal fluid covers your spinal cord as well as your brain, providing some cushion and buoyancy, as well as some chemical stability, because we are going to be able to remove the waste produced by the neurons outside of the brain using the cerebrospinal fluid. As we mentioned before, we have cells in there that are going to make the fluid. Those are going to be the ependymal cells. If we look at the anatomy of our cerebrum, we can see that we can divide that in lobes, and this is going to be frontal lobe, parietal lobe, occipital lobe, and temporal lobe. But we also can divide the cerebrum in two different pieces by this long groove right here called the longitudinal fissure, right? The longitudinal fissure separates our brain into the right hemisphere and the left hemisphere. If we look at the top of our brain, we can see that our cerebral cortex is not flat and smooth like this screen that you have in front of you, but rather it's going to have grooves and elevations. Groove right here, there's going to be an elevation right there. So the grooves are going to be called sulcus and the elevations are going to be called gyres. You can clearly see right here with the sagittal cut of the brain cavities. And these ones are the ones that I was telling you, they're going to be called the ventricles. And right here, you can see the groove, which is the longitudinal fissure. Okay, if we scroll down right here, we can see that our brain is going to have different areas, and each one of these areas are going to be responsible for different functions. The cerebrum is going to be divided in frontal lobe, right? For example, that one, we are going to use it for moving. It's going to be responsible for motor control. This is the parietal lobe. That one is for sensations. For example, right, if your skin itches, you're going to feel that in there. The occipital lobe is for vision. This one right here is the cerebellum. The cerebellum is going to help us control equilibrium. This part right here, we say it's the diencephalon. The diencephalon is going to have multiple different functions. One of them, for example, is going to be control of hormones. And this one right here is going to be the brainstem. The brainstem, as I mentioned, is going to have three main areas: midbrain, pons, and medulla oblongata. This area in particular, midbrain, pons, and medulla oblongata, are going to be related to the nerves that we have right there inside our cranium. So these ones, the cranial nerves, are going to come out of your brain stem. If we scroll down right here, those are the same figures as the ones that I showed you before. Right here, you see the brain stem again, where you have the midbrain, pons, and medulla oblongata. The top part is the diencephalon. The diencephalon is going to have three main areas. As you can see right here, it's going to be the thalamus, epithalamus, and hypothalamus. So we will scroll down right here. Regardless of the areas that we are going to have in our brain, they are going to be protected, we mentioned, by the cerebrospinal fluid, but also by the membranes. These membranes are going to be the meninges. We said we have dura mater, pia mater, and arachnoid mater. The dura mater will cover also these areas where we have the grooves. For example, we said this is the longitudinal fissure, so therefore the dura mater will go in there inside in order to protect that. And these are going to be provide these structures formed by the dura mater called the falx cerebri, falx cerebelli, and the tentorium cerebelli. The falx cerebri is this one right here. Falx cerebelli is the one that separates the two hemispheres in your cerebellum. And the tentorium cerebelli is the one that separates your cerebrum from your actual cerebellum. So we said that these are going to be the three membranes, right? Dura mater, pia mater, and arachnoid mater. The cerebrospinal fluid that we mentioned before is going to be located in the ventricles. But where is this going to be formed? It's going to be formed right here in the ventricles. to be more specific, in the choroid plexus. In the choroid plexus, you're going to have these cells called the ependymal cells, and they're going to produce this fluid. So then we produce approximately 500 milliliters. So then we produce in here 500 in the choroid plexus that we have in the ventricles. And we are going to remove this fluid through the arachnoid granulations, which are these granulations right here. You can see one, you can see another one right there. Those are going to be the arachnoid granulations. How much fluid we remove from our nervous system through the arachnoid granulations? Approximately 350 milliliters. How much fluid we have flowing basically inside our brain and our spinal cord, then we're going to have obviously 150 milliliters, right? We produce 500, we remove 350, therefore we have basically 150 milliliters inside at all times. So now we have in here the autonomic nervous system. We mentioned that the autonomic nervous system is going to be this set of nerves that we're going to have at the level of our spinal cord, but these are going to be the nerves that are going to be the nerves that you have in the thorax. Because remember, we said the ones that we have at the top, these are going to be the cervical plexus. This is going to be the brachial plexus. Right here, we said this is going to be the lumbar plexus, and this is going to be the sacral plexus. Right? Cervical plexus, brachial plexus, lumbar plexus, and sacral plexus. So we said the set of nerves that we have in the middle, which are the thoracic nerves, this one right here, that's what I showed before. These are the ones that are going to be related to the autonomic nervous system. Right? We mentioned before the autonomic nervous system is going to have two divisions. One of them is going to be the sympathetic nervous system. The other one is going to be the parasympathetic nervous system. Right? We said the parasympathetic nervous system is going to be involved in making the different organs and structures to move faster, especially those structures that are going to be involuntary. And the parasympathetic are going to do the contrary, right? The opposite to that, which is going to slow down structures that we have in there. For example, if we talk about the heart, right, the sympathetic is going to make your heart beat faster, right? And the parasympathetic is going to make your heart beat slower. Right? So then remember, the heart is just one example, right? You have also the pupils, right? The pupils that you have in your eyes, right? The sympathetic is going to enlarge your pupils so they become larger, and the parasympathetic is going to make your pupil become smaller, right? So then that way you can see here that heart is involuntary, the pupils are involuntary, therefore those are going to be controlled by the autonomic nervous system. How many divisions? Two, sympathetic and parasympathetic.